Welcome back, everyone. So I hope everyone had a sufficient quick break. I'll welcome you back now and welcome back all of our people online as we settle in for our next session. And it's really, while you guys are sitting getting settled, I want to say a few thank yous and acknowledgements. For this workshop, I really want to acknowledge the CRCG and Professors Jim Pauly and Anna Schwendeman, Dr. Visha Krishnan and Jennifer Dick, and many of the staff here at the universities at Shady Grove. But I do want to give some very special acknowledgement to Professor Audra Stinchcombe and to Dana Hamill, who have also been associated with the CRCG and the University of Maryland. It has been absolutely central, the work that they have done to help and support and make this workshop happen, as well as Bianca Reginald and Sayapavani Gundlapalli, sorry. Thank you to both of them. They were graduate students in Professor Stinchcombe's lab, and they helped to produce many of the videos that you'll be seeing in this afternoon's session. One of the things that we sought to do was to try to not just provide information, training, demonstrations based upon idealized situations, but really to try to identify scenarios that happen in the real world as being particularly instructive to the discussions that we hope we'll be getting into during these sessions. So a big thank you to those individuals. I also really want to give a big thank you to the diffusion cell manufacturers, Logan Instruments, Permagear, and Teledyne Labs in alphabetical order. There's no preference. They're all great systems. And also to the TEWL instrument manufacturers, Delphin and Cortex, who kindly provided their instrumentation as well. All of these manufacturers really have given a great deal in terms of their equipment and their availability to help make today happen for all of you as this free workshop. And then, of course, Dr. Priyanka Ghosh and Dr. Tanaz Ramazanli, who were really central in developing a lot of the curriculum here, along with so many others. But before I say anything further, also a very key member of this entire team has been Dr. Ahmed Zidan, from whom you'll be hearing from. He has led a lot of the work in this area, and I think is going to give what I think you'll find to be a very, very useful presentation, not only today, but as a permanent reference going forward. So it's my pleasure to introduce him as he gives us a talk on designing IVPT methods with some really practical insights for integrating and being able to implement the IVPT guidance recommendations. Dr. Zidan is a senior pharmacologist within FDA's Division of Product Quality Research in the Office of Testing and Research, where he leads the topical and transdermal drug products laboratories and provides training to reviewers in this specialty area, as well as in several others. For example, Dr. Zidan also serves as an expert for the USP on complex excipients. His research accomplishments include an extensive body of publications and book chapters, and he's been invited as a speaker at numerous scientific meetings around the world. We're very lucky to have him with us today. He completed his bachelor's and master's degrees in pharmaceutical science at Zagazig University and his doctoral degree in drug delivery at Howard University. Please join me in welcoming Dr. Ahmed Zidan.
Good morning, and thank you all for joining us in the workshop today. I think after the wonderful first session of the workshop, let's take or move the gear a little bit into more technical parts of IVPT. Let's talk more about the science behind IVPT, science behind diffusion, and how we can develop a test that really can be sound and may be used for a surrogation of bioequivalence. So this is disclaimer of my talk. And let me first ground my slides with this statement from Office of Pharmaceutical Quality that everyone deserves a confidence in his next dose of the medicine. And then our mission and the mission of pharmaceutical quality is to assure availability of safe, efficacious, and this is in every dose of the medication. And of course, semi-solid topical drug products are no different. With each dose of that the patient's taking, it has to meet same level of quality and safety expected for it. So in my presentation today, I'm going to cover first, what do we need to know about IVBT testing? Then I'm going to talk a little bit about diffusion kinetics and what is the theory behind IVBT testing. And then what about the parameters for IVBT in terms of scan, diffusion equipment, receptor media, dose application, barrier integrity function, as we have seen in the first session, and then touch a little bit about validation. And then I'm going to give one of the research that had been done in FD labs showing the feasibility of IVBT testing for demonstration of bioequivalence. So why do we need IVBT? IVBT is usually need for assessment of bioequivalence, and this is a part of NDA submission, as we just heard. And the main scope is that IVBT can be parallel to the PK study. However, there is no any distribution, metabolism, or excretion phase. And actually, the decline that we are seeing in the flux curve after a certain dose application to topical product, it's coming from depletion of this dose. So the dose volume and the amount that's applied to the skin is the key for the flux profile attributes or the shape of the flux curve. So because it is parallel to the PK study, so we are using some of the endpoints analogs, for example, EUC or partial EUC or Cmax or Tmax for comparison. And more prominently is the issuance of the IVBT guidance that just released. And in this guidance, it outlined most of the critical steps that we need to consider if we want to develop IVBT study. So, but before we develop the study, what do we need to know? I believe the first step is to understand your drug product, right? This drug product about API characteristics and its contribution to the microstructure. What is this microstructure is formed of? What is the critical quality attributes that manage the drug distribution within this microstructure? When we are applying a certain topical formulation to the skin, so drug release is happening, drug permeation, drug partitioning, but it happened in a dynamic way that's maybe different than what's happening in IVRT or IVBT testing. So we need to understand more about the matrix, about the microstructure of the drug product and how it manages the diffusion kinetics. And the second is about our diffusion equipment. So what is composed of? What do we need to consider to develop an IVBT test in terms of selection of the testing parameters? And then at the end is the validation. Is the test is really valid to discriminate or show similarity between reference and test product or not? So we're going to touch base about validation. So for this one, I'm starting my my presentation with a very big slide where it's showing all the IVBT parameters that you may think of. These parameters can be categorized into a number of groups. First group is mainly related to the skin itself, as the skin sample, starting from skin parameter, source of this skin, and is it thickness of the skin, is it fresh, dermatomed, etc. And then skin storage. Are we using fresh skin or it is or it is like frozen for some time? What is the storage procedure that had been used? Are we including any cryoprotectant so that the ice crystals does not interrupt the skin barrier? As well as how we are measuring the skin barrier function in terms of measuring the water loss or electrical resistance or even diffusion of treated water or radioisotope water, right? And then another group of parameters that's mainly related to our equipment, the diffusion equipment, diffusion cells, right? So what is this type of diffusion cells? I think the gold standard that we are using is vertical diffusion cell. However, many applications are using also flow-through cell, What are these parameters? What are these cells are made of? Is it borosilicate glass or is it Teflon? What is the donor cell is made from? How they are stacked or assembled together? And then what about the diffusion cell parameter itself? Like the diffusion cell parameter, like the diameter of the diffusion cell, exposure area, mounting the skin, and all other criteria. And what about the diffusion kinetics? So what is the lag time for diffusion? Do we need to consider this in the method optimization, as well as like what is the flux, how when we reach the maximum flux from after a certain dose? And then more factor related to the receptor solution. This receptor solution is to maintain same condition, maintain solubilization. So what is the composition? What is the pH, solution osmolarity, or isotonicity if we want to maintain? And how we can optimize the receptor media. And then some, I would say, discrete parameters that's related to the experimental condition for diffusion, temperature, steering rate, or removal of the drug that's permeated from the skin surface so that it maintain, like I say, unidirectional flow or a steady state flow of diffusion. And sampling schedule, for example, what is the sample parameter? We just heard some discussion about like full sample or full media replacement or aliquot sampling, which one that's more robust. And then data reporting, how we are reporting these data in terms of flux or cumulative amount released. Do we have enough time or duration for it? And then many factors that's related to the bioanalytical method that you are using to analyze these samples. So we just heard from industry that there may be some challenges when to detect the drug in a very minute amount. In some study, it can reach to picogram amounts. So do we have a really efficient and validated bioanalytical method to analyze these samples? So what about the suitability of the method? What about the validation of the bioanalytical method away from the validation of the IVBT test itself? So this looks very, very comprehensive. So do you think we can control all of this? Do we need to think about all of this when we are developing the method? Let's talk about the real scenario here in any of the labs, in FDA labs or any of the company labs or any of testing labs. We are starting with what we have, right? It's not essentially to start with a certain instrument, not essentially start with a certain parameter, but there should be some point to start from. And then there is no only single way to get to a successful IVBT testing or a good IVBT profile, but there is a way to have a controlled method that can give you an acceptable flux profile or IVBT testing. So for this one, I will start with, if you want to develop a method, what you need to do. So first, you need to have a comprehensive summary table. This summary table should have like source of skin, for example, what's harvesting method, preparation of this skin and mounting it, skin origin, does it coming from cadaver or fresh skin or anatomical site, suitability of the donor, for example, age of the donor, gender, condition of the donor, equipment selection, like are we going to use like vertical diffusion cell or flow through cell dimension, size of the steering bar inside, and then receptor solution, composition, volume, and then diffusion parameters there. What is the method that you are going to use for skin integrity, and what's your acceptance criteria for this for skin integrity testing? Dosing parameter, how you're going to apply the dose, and how you are ensuring that you have a robust and consistent dose application to all skin samples. Then you are going to collect your sample over a certain duration, how you are going to purify it so that it becomes suitable for the bioanalytical method, and then how you are ensuring that there is no interference of this. So in order to put the parameter in this table, let's talk a little bit about the diffusion kinetics. So as we heard from the previous session, most of the skin integrity testing that we are using, it is just to measure, I would say, integrity of skin structure, but not necessarily it reflect the barrier function of skin for diffusion of a certain molecule from a certain formulation. So we are using this model that is, I think, proposed by Franz and Lehmann in 1995, where we found that the flux, sorry, yeah, okay. Yeah, I'm not sure if you can see it. So if we look at this flux model, the flux of the drug through this membrane function, it is a function of permeability coefficient or permeation coefficient and diffusivity or diffusion coefficient. But most important, that's more related to our development work, is the concentration of the drug here, right? So the concentration of the drug is directly proportional to the flux. As you are applying more drug to the dose, then you will have higher flux until this dose is depleted. And then you may get two shapes of the curve. If the amount of the dose is really large dose, so it is infinite dose, then you are going to have like I would say sustained flux. And in this case, the flux will give you us a steady state phase. And this steady phase will start to decline when the dose is depleted from the donor, or if the same condition is not met, right? Because it's large amount of the drug permeated. And when we're applying a finite dose, then we are going to have like a sharp increase in the amount that received in that found in the receiver media, and then we are going to have this peak or flux peak or Gmax that's comparable to the Cmax that we are seeing in the in vivo. So it's not necessarily to find that this Gmax is reflecting steady state. However, it's merely reflecting the depletion of this dose from the media. So as much dose that we are adding there, we need to avoid going to the steady state, or if we go to the steady state, then we need to find a way to decrease the steady state to see the depletion zone by wiping off the drug or the dose from the donor cells, or even like to use a smaller dose. So in this case, understanding the diffusion kinetic here will help us to develop or to set what would be the starting point for the diffusion parameter. So the first step for us is the selection of diffusion equipment. So I cannot say that there is a preference of one setup of diffusion, like an instrument, or I'm sorry, equipment compared to the other. However, the main understanding is that we have a donor compartment, and then we have a permeation media, which is the skin, and then we have a receptor solution. So we just need a setup that's showing a direct flow, or I would say steady flow of the drug through from the donor across the permeation media, which is the skin, to the receptor solution. So I think the very nice general chapter of 1724 in USP is a very nice start of selecting which like setup for diffusion testing you may be using. And this is have all qualification test and component of this receptor media. And many thanks here to the manufacturer who came to show us here like all the different types of diffusion set available, like from Logan, from Teledyne, and from Permagear. Second parameter that we may talk about is the selection of receptor media. Which media to use? I think the first criteria for the media here is the compatibility. So this media that you are using should be compatible with the skin barrier. So don't include any, I would say, components in the receiver media that may affect the barrier function of skin. So for example, organic solvents or certain type of lipid that may make perturbation of the lipid structure of the skin. Second is the drug stability through IVBT sample, and even during the samples that's collected from IVBT until analysis. So we need to ensure that the drug is stable in these samples until we are getting the data from it. So we need to make sure that there is no interference in the bioanalysis. And the most important is the solubility requirement. So we need to select a receiver media that's showing, I would say, acceptable solubility of the drug so that sink condition is met. And this is mainly achieved by inclusion of solubilizers, because we know that most of the drug applied to the skin is hydrophobic drug, and we may use solubilizer to facilitate the same condition. In this case, the guidance is suggesting some solubilizers, but this is not, I would say, only solubilizers to be used. Mainly you are using like OLIS 20, we can use it from 0.1 to 6%. And this surfactant is known not to like interrupt or affecting skin integrity. However, you may use other solubilizers justified by data that these solubilizers are not affecting like skin integrity. I'm sorry. And for a study that may go beyond 2 days or 3 days or 96 hours, then you may justify using of additives like, for example, antimicrobial agents, sodium azide or gentamicin, to ensure that there is no microbial growth that may, I would say, like degrade these skin samples during the test. And other additives may be used like stabilizers or pH modifiers. And we need to ensure also that the receptor media is allowing like sensitivity assessment to higher drug permeation, which is needed for the validation part. And then what is the volume of the receiver media to be used? As we are going to see in the instrument demonstration, and all of us have experience in the lab, we have many sizes of receiver chambers that can take like large amount of media, and even we may use flow-through cells, which is using like large amount of the media. So it is a balance between solubilization and analytical capability of your method to detect the drug. For example, if the drug is highly permeable from the skin, and then we are using large volume receptor in vertical diffusion cell, in this case, same condition are maintained. But if the drug is highly permeable and we are using a small volume of the receiver media, so this will be precipitation and non-sink condition, and this will affect your steady state and will affect the flux peak. And if the drug is low permeable and we are using large volume of the media, then of course there will be some bioanalytical challenges there. And it is the same for the flow-through cell assembly. As the drug is flowing under the skin or at the dermis layer or at the dermis surface of the skin, if the drug is very highly permeable and then we are using high flow rate of the receiver media behind the skin, we are collecting large volume of the receiver media, and it may represent some analytical problem here. And if the drug is low permeability and we are using high flow rate, then of course analytical problem will be there. And then if the drug is low permeable and you are using low flow rate, then it's better analytical detectability. However, same condition may be a question, right? So all of these characteristics has to be like considered for selection of the receptor media. So what about those application? So there is many ways to apply the dose during IVBT testing. But most important is to find a way that's applying a consistent dose among all replicates and for all the runs and between the test and the reference product. So we usually use a positive displacement pipettes, as you can see here. And this positive displacement pipettes ensure that a certain volume is delivered so that a target mass of the product is delivered to the skin. And usually this is corrected based on the area of skin to be dosed. So it is normalized per centimeter square of application. And then we need to make sure that this pipette is calibrated and reproducible for a certain test. So for the pipette calibration, you know that we are using semi-solid product here, which is very viscous, and the delivery and suction in the pipette tips or the positive displacement pipette tip, it may be challenging to be delivered, to be withdrawn. So we need to, like I would say, we need to validate the pipetting method and that it is fitting for the purpose. The most important parameter here also is the dose spreading and standardization. So there is many ways to spread the dose. Some of the ways is to just use like HPLC vial, and the dose is applied at the bottom of the HPLC vial, and then it is inverted, dispersed like over the skin, and then reweigh it again to ensure that it is the same amount delivered. Another way, just by the positive displacement pipette, you just apply a certain dose, and you may use a glass rod to spread this dose over the skin, and then the glass rod is reweighed again to ensure that it is, like I would say, consistent dose is applied to the skin. So residual product consideration has to be considered here, and the method reproducibility should be established as a part of your method development steps. Skin sample, which is the key player in IVBT. So there is many parameter and many factors that is we are accusing or we are trying to relate it to the skin. That is, skin is the one that's managing all the errors. However, there should be some inclusion and exclusion criteria and standardization of selecting, I would say, suitable skin samples for our test. So many factors coming from the skin and may be responsible for the variabilities that we are seeing. So we can summarize it in this table where the type of skin, source of skin, and supplier of skin, and the skin quality, how it is dermatomed, for example, and skin penetration, and skin tracking system, ethical and regulatory consideration. So let's go over this one by one, which is because it is very important part of development. So different types of skin samples may be used for IVBT. For example, full thickness skin, where the epidermis, dermis, and subcutaneous layer may be used, or dermatomed skin, which is many of us are using, or only epidermis skin, which is split thickness, and this epidermis can be removed by heat separation or even by enzymatic activity, for example, to remove it. But more processing of the skin is more variation in the skin condition. So another parameter in the skin is the source of skin. Where did you get your skin from? So we may get it from a surgical waste, like from plastic surgeries, or we get it from the cadaver skin, we're getting from skin banks or biorepositories. And also the suppliers of skin. Is it commercial skin supplier who have enough knowledge to handle the skin and standardize the skin condition during dermatoming, for example? And this happen in the tissue procurement organization or biobanks that specialized in handling like human skin, or it is done in-house collections, right? Like, for example, when you have a research institution and hospitals, they are integrated together to collect all samples that, or skin samples that's generated. So the laboratory collection of skin, it needs certain skills to standardize the thickness and to make sure that this skin is healthy enough for the test, whatever its source of coming from. Skin quality. What about the skin quality? Donor characteristics may affect the skin quality and its usability for a test. For example, the age of the donor, sex, is it male or female, and ethnicity of the donor. We know that more melanin at the skin surface, it may interrupt or may impact the drug absorption, and also healthy state of the donor. Is it disease or healthy skin is collected? Anatomical site of skin, is it taken from the forearm or the back or abdomen or thigh? Is it hairy region or glabrous, like non-hairy region? Because more hairs in the skin, it can affect the flux and permeation, as well as the thickness of this skin, what is the variation may cause to it. And how we are how are going to measure the skin integrity and suitability for the test. So skin tracking and database management. So we need to have a system that is like associating skin samples to a certain test from a certain donor. So usually we are using unique sample like that has the donor ID, tissue type, and date of collection. Sometimes we use barcoding system for tracking, and this barcoding is linking all the metadata from the donor, his medical history, and it's like information that may be useful for the test. And we need to keep a system for record keeping and database management. Sometimes we are using a laboratory information management system, and then that's including all information about the storage, retrieval of this system, and then data sharing policy should be considered. And then storage of the skin. So skin, as it is procured from the source, it may be used fresh when it's used within 24 hours of collection, or it may be snap frozen in liquid nitrogen before it is applied to certain test, or it may be stored at -20 to -80 degrees for a long-term preservation, as it may affect the skin condition. So is the supplier is including any cryoprotectant, for example, like are they using glycerol to make sure that the freezing process does not affect skin integrity, and as well as the thawing and rehydration procedure, like is the thawing, as we have seen from previous talks, like the thawing temperature and equilibration, is it affecting skin integrity or not? And what would be the rehydration liquid to use in order to thaw the skin? So skin preparation techniques. So is the skin thickness standardized or not? Usually dermatomed skin is collected by professional people, but in some places they are not professional. You may have some variation in skin thickness, which is one of the criteria to be used, like is this skin is suitable to be used in these replicates or not. And then we need to verify the skin thickness using, for example, calipers or optical like coherence tomography. And the procedure that we are using to clean the samples, are we are using like scalpel, or we are using like enzymatic digestion to remove all the fats or the subcutaneous tissues. And then we need to avoid any mechanical damage or insults to the skin so that it does not affect skin barrier. Also about the hydration. So we need to standardize the hydration process or hydration procedure for all skin samples, and we need to avoid overhydration because it may affect the skin barrier function. And if it is overhydrated, then another equilibration time, or I would say treatment for the skin to make sure that skin is still suitable for the test. So what about the ethical and regulatory consideration? Do we need to have in place like an institutional review board, IRB, like protocols to be submitted to ensure that all ethical requirements are in place? And this is a check the storage and disposal according to biosafety guidelines. And this is in compliance with the safety requirement by many institutions. And is there is any need for a special consideration for IVBT? For example, is the drug is hydrophilic or hydrophobic, or do we need to have a certain requirement to have the test fit a certain application? Do we need to compare it with a certain, I would say, another barrier or in vivo barrier, or do we need to cross-validate it with biological barriers? All of this has to be considered for the skin in order to, I would say, qualify the skin for the test. One of the very important parameter here is the skin integrity. So skin integrity here, it is a test that's showing that the epidermis or the stratum corneum part of the skin is of the same structure, or I would say not affected by the procedure. One of the gold, I would say, skin integrity test is the transepidermal water loss, or as we call it, 12. This one is just measuring the water flow or water vapor flux out of the skin. Water is polar, right? So because the water here is diffusing through the skin, it not necessarily reflects the barrier function of skin. But we are measuring that as the stratum corneum exists like at the skin surface, so it prevents a lot of water to come out of the skin. And in this case, we need to set a criteria or acceptance criteria for this water loss. Usually, we are using different tool systems to measure this water vapor loss. And these systems have different designs. It can work as open chamber, where the sensor for the humidity is just at the end of the chimney for the system, but the system is open at the skin. Or it may be closed system here. I'm sorry. It may be closed system here, where there is no like further evaporation of skin is happening, or it may have a condenser here where it allows condensation of all the vapor, and this is used for longer time. So we want to say that this tool measurement is depending on the device that we are using, the operation, and the environmental, I would say, around this device, mainly like the use of the adapter and how far is the sensor from the skin surface, and precise control of this experimental condition like the temperature, humidity, probe alignment, and sensor distance, and the adapter, and the hand heat transfer. All of this has to be controlled for tool measurement to standardize the test. And we need to have at least 3 replicates per each sample, and setting criteria as it is recommended in the guidance, like below 15 gram per meter square per hour is a standard. However, lower threshold may be recommended in order to have, I would say, more closer condition of skin or more acceptable skin condition for the test. Failed skin sections for tool measurement can be excluded, and regular calibration of the instrument may be used. So another method to use for skin integrity testing, I think we heard about the tritated water skin barrier integrity test, where we are using tritated water here to measure its diffusion through the skin. So water permeation through the skin does not reflect the actual drug substance or drug molecule diffusion through the skin, right? But the test setup is just by assembling the vertical diffusion cells with the donor and receptor chamber, and then applying a small dose of tritated water, and then measuring the amount of this tritated water that will diffuse to the receptor media. So, and this is, we can use scintillation counter to measure the equivalent volume per centimeter square that's diffused. The acceptance criteria, as it is recommended in the guidance, it is approximately 1.5 equivalent microliter per centimeter square. So any, I would say, measurement more than this value will indicate that there might be a problem of skin integrity or the barrier function of skin. And in one of the studies that FDA have co-authored before, that we have measured the tritated water diffusion across different days of an incubation with the skin, and we found that the healthy skin is just maintaining this diffusion of the water over 3 days or 4 days, and most of the diffusion flux values was about like below 1.5, and this from where this number was recommended. So we just don't forget to like normalize the tritated water flux per the surface area of skin that is permeated. So another test for the measuring skin integrity, it is the one that we just discussed, is about electrical resistance of the skin. So if we are adding any ionic substance at the skin, these ions may diffuse through the skin, like from the, like I would say, tight junction or around the tight junction of skin. So we can measure the resistance for passing or flux or flow of this ionic material across the skin. So usually, if the skin is healthy enough, then we are going to have high resistance for the passage of ionic substance. But if the barrier function is interrupted or it is not maintained, then we are found that the ionic substance is just passing easily through the skin, and then the resistance will be less. If there is a certain value for acceptance criteria for the transepithelial electrical like resistance here, actually, it depending on the instrument that we are using, it depending on the measurement, the setup that you are using for this test, but it has to be justified, and it must be validated by experimental data to ensure that they accurately identify a compromised versus healthy skin for the test. The test here can be variable because depending on many methods, like for example, the instrument that we are using for measuring the transepithelial resistance. But in any way, the test is sensitive to this instrument and measurement frequency and other procedural condition. So there is no any preference which among these 3 integrity tests to be used, but most important is to standardize the measurement procedure and to make sure that it is suitable for the test. So the consideration mainly for skin integrity, it is a balance between the acceptance barrier function, what is the steps to be taken to ensure that we have an acceptable barrier function, and what or how we can set the inclusion or exclusion criteria for this test. So for the acceptable barrier function, we need to make sure that the handling procedure and skin preparation is not affect the barrier function. For example, the hydration change in the skin, we need to allow the skin to re-equilibrate again so that it maintains the barrier function. And we need to avoid any external factors that may mislead the judgment from the test. For example, temperature, environment, humidity, heat from the lamp. Sometimes we are using lamp very close to the skin that allowing more water to diffuse out from skin or evaporation. And this is also will give us misleading data here. And also functional discrimination of barrier function. We need to make sure that the test result here can distinguish between intact skin and the damaged skin that's damaged intentionally by using like any insulting in the lab, for example, by tab stripping, where it's removing the stratum corneum. So we need to make sure that the test results can discriminate between intact and damaged skin. So when we ensuring the acceptable barrier function, we can easily set the acceptance, the inclusion and exclusion criteria based on these acceptance values. So the acceptance values here has to be data driven. This means that the values that we are using for each of these tests has to ensure accurate identification of compromised skin from a healthy skin, and only excludes the compromised skin from the test before you do the permeation testing. And we need to make sure that the test is sensitive enough so that it can differentiate between damaged and undamaged skin. Okay, so I would say the last parameter here to talk about is the study duration and sampling schedule. So the study duration, it is mainly a function of the flux profile. And what is the attributes of the permeation profile that you are looking at? So I'm looking at the J flux or the maximum flux peak. I'm looking at declining of the applied zone due to depletion from the donor chamber. So in this case, we need to extend the study in order to cover these features so that we can detect the flux peak and detect the decline. However, this is not necessarily to be achievable in all formulation, because at the end of the day, it is determined by the flux from through the skin and the drug release and partitioning with the components and the metamorphosis events that's happening to the skin. But most important, just make sure to document all the effort that we are used to optimize your ideal profile. And this is a profile that we have gaining from this one. So once you find a declining from the flux curve, you may consider shortening the time for the test, maybe from 4 days to 2 days or to 1 day, as long as you are getting enough features that you can be used for comparison of permeation of test and reference product. And then refine sampling schedule. How many samples do we need to collect? So the samples need to be collected based on these features. So we need to collect more samples around like the flux peak here. So if we go to most of the guidance or I would say like the practice that we're using for PK study, we used to collect at least 3 samples before Cmax, and we collect the samples up to maybe 4 or 5 T-halves of the drug after the Tmax. And this one shouldn't be different, which is showing that we have enough sample to define the peak of the flux or the maximum flux here, and we have enough samples to see the decline of this flux, right? And then this is need to be aligned with an efficient bioanalytical method that can detect the drug precisely and like robustly. And make sure that you identify that whether the samples are analyzed with a validated method or not, whether the cells are run in parallel, and whether we have a balanced study where we are just like randomizing the treatment per skin samples among the donors. So this is what we started with in the first slide. We mentioned in the first slide that you need to have a table or a summary table of your method development. This one is just example of this table, which is just showing what is the equipment to be used, which is the one available in your lab, the formulation, strengths, applied dose, and the skin samples to be used. And this is just some description of skin samples. Is it coming from human cadaver skin? Is it dermatomed? Is it full thickness skin? Donor is male or female, and it is like what is the age of the donor, thickness of this skin, receptor solution, what type of receptor solutions that we are going to use, what type of integrity tests that we are going to use, and then receptors volume, and then sample processing, like if we are going to need to have a purification method, and then skin temperature, the steering rate that we are going to apply for the test, and the orifice area for permeation, and sampling time point, length of the test, and sampling to be collected. What is the sample volume? Are we going to allocate some of these samples for priming or washing the lines? And then we are going to run the test under occluded or non-occlusion condition, and then the donor, number of donors to be used and number of replicates per donor. So this should be our starting point for the test. So once we have a good permeation profile that give us, I would say, feasibility idea about the permeation, we can go for the validation, which is moving from the pilot study to pivotal study. And in this case, we need to conduct a pilot study first in order to make sure that the test is suitable to show the similarity or discriminate between the test and reference product. And we need to ensure sensitivity and selectivity. And this will be covered extensively tomorrow in the fourth session. And we need to ensure we have adequate statistical power. The number of donors, as Dr. Samrani showed in his presentation, the number of donors to be used for certain tests, and number of replicates to be used for each donor. And then we need to ensure that we have a balanced study where a randomization of these skin samples among the test and the reference product is considered so that we are avoiding any bias there. We need to make sure that we are separating the data collected from the pilot study and pivotal study to avoid any bias. And we need to ensure that we are following protocols and we are setting protocols for equipment like qualification, suitability, and methodologies to be used. And we need to make sure that we have a controlled IVBT run so that there is no bias over the test. So we can move now to one of the examples where FDA have used in FDA labs. We just test the feasibility of this test for IVBT. So our drug product here is ruxolitinib topical cream. This ruxolitinib here is used for atopic dermatitis. It's used to treat eczema, and it is formulated as oil and water emulsion. And it exists in two strengths, 0.75 and 1.5% strengths. And it is manufactured by like blending and homogenization process. So if you remember in my first slide, when I told you what you need to know for the method development is to understand the product. And this is what we are going to mention here now. What is the microstructure of this product? So this product is oil and water emulsion cream, meaning that you have a matrix of oil droplets stabilized by surfactants, and it is dispersed in a continuous phase. And depending on the solubility of the API may be partitioned between the oil droplets inside, like in the oil phase inside the oil droplets, and around the micelle structure, like in the continuous phase, and even solubilized in the outside or in the continuous phase of the drug product. All of this is interacting with the skin, and the dynamics that's happening due to depletion of the dose at the skin surface is there is some dynamic that's happening here where the drug is diffusing from the droplets to the aqueous phase and coming to the in contact with the skin for permeation. So depending mainly on the partition and physicochemical properties of the API, we may need to consider this in developing a method. For example, ruxolitinib here is a highly permeable compound. However, it is used as a salt, which is ruxolitinib phosphate. It has a molecular weight of about like 400 gram per mole, which is favorable. I would say it's large, but it is allowed to like passing or crossing the skin. And the ruxolitinib has a pH-dependent solubility. Because it is a basic drug used as a salt, so it has high solubility at lower pH, and solubility declines so fast when the pH exceeding 8. The ruxolitinib base itself is soluble in methanol and ethanol and water. And I just give you like hint about the drug distribution among formulation. If you wanted to develop IVBT test for this drug product, the first part I would do is to test the solubility in different receiver media. So we can select, we mentioned the parameter to select the media, but I will just highlight the solubility of ruxolitinib and ruxolitinib phosphate in different media that we know that it does not affect the skin integrity. So we tried first like phosphate buffer of pH 7.4. And because the solubility of ruxolitinib base is low in aqueous media, so we tried to include some, I would say, solubility modifiers or solubilizers like 0.2% OLS 20 and 4% of bovine serum albumin here. So we find that the phosphate like salt is soluble in all of these media, and its solubility does not affected by OLS or bovine serum albumin. But the solubility of the base is mainly affected by the surfactant or bovine serum albumin. So we can say that most of these three selections may be used as a receiver media, which is guiding the development of the IVBT study after that. The first run of the IVBT study, it's including the using of FINIS, dry heat diffusion cells, and we applied a dose of 11 milligram per centimeter squared, and we just screened these three media to see the profiles. So in the first run, we have used just phosphate buffer, BBS. Second, it was phosphate buffer plus OLS. And the third run, we included phosphate buffer with serum albumin. We included sodium azide here, as antimicrobial, added to the media. And then we did a skin integrity test using water loss or transepidermal water loss. And the receptor solution was 15 ml here. Sampling schedule, it's up to 48 hours. And the duration of the test is 48 hours, of course. The samples collected, it's about 700 microliter. 500 microliter is collected for sample analysis. And the 200 microliter is used for priming the test. And we run this test under non-occlusive condition. And we have done this test from skin sample from one donor at 3 replicates per each. So we found that the drug was almost the flux was almost similar in phosphate buffer or phosphate buffer that has OLS 20. But we start to seeing drug in the samples after 8 hours. So we're actually missing the first part. We have a long onset here for the drug to be permeated. And when we used bovine serum albumin here, we didn't find any improvement so much of using bovine serum albumin. However, we found that the flux is even more retarded. The first drug was seen after 48 hours here. And we concluded that adding OLS 20 or bovine serum albumin does not provide more advantages than just using phosphate buffer for this test. And then we decided we are going to use phosphate buffer to continue the test here, as long as phosphate buffer is providing the same condition for all the dose to be released and depleted from the donor chamber. So the further optimization, it included increasing the dose from 11 milligram per centimeter to 15, and then decreasing the receptor volume from 15 ml to 6.5 ml. And this is necessitates the use of other instruments. So in this case, we have used the Microt automated system. And then we started to find, oh, there is some permeation here. And it's starting from initial time points. I'm sorry. Yeah. Okay. So we'll just see here. Okay. So we can, okay. So we can see here that there is some permeation that's happening here, but we didn't find all the features of the maximum flux, right? The drug is keep permeating up to 48 hours here. So we may say that, okay, we need to optimize the study duration, or we may consider smaller dose, right? However, one of the observations that we have seen here is that in one of the replicate, there were some dooming that's happening to the skin, and it's affecting skin barrier here. So this dooming of the skin here, it's one of the source of the outliers. Yeah, you can see here. So this is one of the source of the outliers, and because this dooming here is exerting physical pressure to break down or to interrupt the barrier function of skin. And this was one of the reasons to exclude this data point from the data set. However, for the other replicates, it was acceptable replicate. So in the subsequent run, we tried to optimize the shape of the curve. So we extended the study to 96 hours, and we increased the sampling schedule. And then it is the same system that we have been used, same dose that applied, and same volume of receptor media. And among we have used in this case 3 replicates and 3 donors, and we have used 4 replicates per each donor. We find a nice, I would say, permeation profile. We didn't see the distinguished flux peak here or maximum flux, but we find that after 96 hours, the drug concentration start to decline here. So we are saying that this is a nice profile. Further optimization has been done using the initial Phoenix system and using the same condition, using another 3 donors and also 4 replicate per donor. And then we have a nice replicates among I'm sorry, we have like a nice profile among these replicates in all the donors. So this study showing that it is feasible. It is a systematic way to develop the permeation testing. So the takeaway from my presentation that developing your IVBT method always start with your understanding of the physicochemical characteristics of the API and microstructure of the product. We need to tailor the method parameters to fit a certain topical formulation and study objective, and select a suitable diffusion cell or diffusion equipment for use. What would be the barrier test function to be used based on availability in your lab? What is the acceptance criteria for the barrier function test? Determine your study duration and sampling interval in order to capture all the features from your flux profile. Determine how many donor and how many replicates to be used for to have a discrimination power. And do you need to think about the validation criteria? And I think we're going to hear more tomorrow about the validation criteria. So my 2 cents here from the presentation is a fit of purpose IVBT method. It is the one that can provide confidence in your topical bioequivalence assessment. So with this conclusion, I want to acknowledge all my colleagues from like Office of Pharmaceutical Quality and Office of Generic Drugs for all of their collaboration over the last, I would say, 12, 13 years, developing IVBT samples, having many like tests of many drug products. I want also to acknowledge all the manufacturers for their support during the workshop, and our research collaborator, and many of the ORISE fellows and contractors who are helping us all over this study, and of course CRCG and University of Maryland in Baltimore for their help with the workshop. Thank you.